is specifically talking about the nucleus. And in Unit 1, we talked about DNA, and I talked about how DNA has the instructions for how to run the cell. It's got the instructions specifically to make proteins. Well, we store that DNA in the nucleus. And then ribosomes will then eventually take a copy of that DNA, we call RNA, and then make the proteins from that. But I'll show you a little bit more what I mean in the coming slides. But point is, the nucleus, where the DNA is at. And I want to go over the structure of the nucleus. Kind of I don't know if there's a lot going on with it, but it's really, really interesting. So the nucleus, when you're like in freshman bio, it's kind of like it's this big sphere. Just remember that, I guess. Well, we want to start getting you to learn more about the structures. The nucleus has a lipid bilayer, kind of like the cell membrane does. But you've heard of double rainbows? How about double membranes? The nucleus is it has two lipid bilayers. So you have an outer membrane, and then you have an inner membrane, and collectively we call that the nuclear envelope. Okay, so that's kind of that zoomed in there. So notice how the circle there, that's the hydrophilic heads there, and then the tails. So that's a really good test question, right? People like to ask about that. Students forget it's a double lipid bilayer. And then there is an intermembrane space that you'll sometimes see labeled there as well. And there's nuclear pores that act almost like filters. So it's interesting because you want to control specifically what goes in and out of the nucleus, because you have the DNA in the nucleus, right? DNA is like the most important thing in that cell. You damage that DNA, that cell's toast, right? So you want to protect what's going in there. So that's where the nuclear pores come in. It's super interesting. Like the details of nuclear pore, it is this crazy complicated filter as far as like the size and charge of things that can go in and out of it. It's really interesting. They zoom in on the nuclear pore right here. The pore complex is basically a ton of proteins that do that filtration process I was talking about. But it's pretty cool stuff. Other things, chromatin is just DNA that is unwound. So chromatins, whenever we're going to start, if you remember like PMAT, like prophase, metaphase, anaphase, telophase. So in interphase, this would be a cell in interphase where the DNA is all unwound like a bowl of spaghetti. That way, that's a sign that the cell is actually using the DNA to make the proteins. Whenever it's time to do cell division, that's when you wrap up the DNA into those X-shaped chromosomes that you're used to thinking of. But typically, DNA is actually unwound. How do we wind the DNA to make it into the proteins, or to make it into the chromosome, or even to make it into chromatin? We use these things called histones. So histones are these spiky-looking things. And I compare it to like a laptop charger. Like if you have an unwound laptop charger, and we have 46 copies of DNA, 46 chromosomes. What would be the issue if you just had unwound DNA in the nucleus? What's that? It's gonna get tangled, right? It's like trying to put 46 laptop chargers in your backpack. Not a good idea. So we wind your laptop charger around the central converter. What do they even call this? Like, what is the point of this thing? I don't know. The brick thing, Power brick, the power brick. The histone is like the power brick, right? We wind the DNA around there. So if you wind it just a little bit, it's chromatin. If you keep winding the DNA and keep condensing it, that's when you get to the formal, really wound up chromosome. Anyways, then on the outside, keyword is outside of the nucleus, you see these studded things, these brown spheres. Those are ribosomes. And I mentioned the ribosomes make the proteins. You'll see ribosomes in two spots. You'll see them on the outside of the nucleus, which the nucleus then directly connects to the endoplasmic reticulum, which I'll go over that in a little more detail in the coming slides. But the endoplasmic reticulum basically transports stuff, would be like the simplest definition. There's a rough ER that has the ribosomes on there. Those can then make proteins. But you also will have ribosomes just floating around in the cytoplasm that can make proteins. Last, two more things with the nucleus. One is the nucleolus. So the nucleolus like literally means nucleus within the nucleus. And this is where the ribosomes are made. So nucleolus, you're going to make ribosomes. Makes ribosomes. And then it's not labeled in this picture, but basically like all throughout this nucleus are these like cytoskeleton-like things called the nuclear lamina. That's not right. Lamina. Nuclear lamina are these like protein fibers. These protein fibers that give the nucleus structure and strength, right? Because you got to remember the double membrane for the nuclear envelope. That's just basically a double fat layer. Think of a soap bubble. That's really basically all a membrane is, without any proteins to support it. It's just like a bubble. Bubbles are very fragile, right? So we have all the nuclear lamina, all these protein fibers that run throughout the nucleus, and that's going to help give it some more structure and support. Questions? I know there's kind of a lot going on in this picture. Okay. Let me make sure I set everything here. Yep, nuclear envelope. Again, nuclear envelope has that double membrane. It's the most conspicuous organelle. That's a classic thing you look for to tell a prokaryotic cell from a eukaryotic cell. Remember, pro means no. Thought I'd get more play with that. Pro means no nucleus. There's a nuclear lamina that I was telling you about. All those protein filaments would be a better word. But basically these protein fibers that run throughout the nucleus to give it some structure. DNA is organized into those chromosomes. Chromosomes are the X shapes. But then if they're not quite as condensed, that's when you get to the chromatin. And then again, when the cell wants to prepare to divide, chromatin condenses into those nice formal chromosomes. Nucleolus, that's where you make now they say ribosomal RNA. Ribosomal RNA is the ribosome, essentially. The ribosomal RNA will do some more like folding and some more processing happens to it, but it ultimately becomes the ribosome. Okay. Let's talk a little bit more about the ribosomes. So the ribosome has two subunits. That's an important thing to make note of. There's a large subunit and a small subunit. If you study biology in a little more detail, you'll learn about how like the ribosomes that are in eukaryotic cells are a little bit different from prokaryotic cells, and they'll give like little numbers. I believe it's like S something. It has to do with basically like the density of the ribosome. Whatever, like you can worry about that as you get into further classes. Just know there's two subunits, one large, one small. And then whenever the ribosome is going to make the protein, they come together to read that messenger RNA. The messenger RNA was the copy of the DNA gene, the instructions that we're using to make the protein. When we go over protein synthesis, I'll tell you more about them, but really make note of the two spots for the ribosomes. First spot is they're just floating around in the cytosol. Remember, cytosol is the cytoplasm, just the watery, almost jello-like stuff that the cells float around in, parts of the cell float around in. And then the rough ER. It's called rough because the ribosomes are bound to that part of the ER. If I go back a couple slides, this was the rough ER. Again, they call it rough because if you imagine rubbing your fingers across it, it'd be like rubbing on a studded belt as you feel those bumps that are the ribosomes. Okay. They call them protein factories made out of ribosomal RNA. And there is, as I said, there's a little more processing that happens with the ribosomal RNA. Like there is some other proteins that get added to it, but the basic idea is it becomes the ribosome. And then because you find ribosomes in two spots, like in the cytoplasm and bound to that rough ER, there's two spots where you can make proteins, in the cytoplasm and on the rough ER. Okay, that's 4.3.